I don't think I've tried that before from the breakout room itself, because I only have 21 people here. So let me go to breakout rooms. Where are they?
Then you're left with one breakout
room. Here we are. I got a new menu. Yeah, I'm left with a few of them. They're all going to close in 30 seconds. Okay. So we'll just give them that 30 seconds in case they're talking still. So we're back, at least. I hope we're all back because I bounced around in the breakout room. So the first group A, they're dealing with the topics of red blood cells. What do they look like? What are their structure? Why are those good functional attributes for a red blood cell? And I mentioned last time they are kind of one of the, that and hemoglobin, sort of one of the things that just amazes me in nature. And then you'll go into hemoglobin, and finally we'll talk a little bit about fetal hemoglobin. So who is the spokesperson for this first group?
I am, Professor.
Okay, Manri, go for it.
Okay, so for question 3, we decided to go with the red blood cells are small in size. They're just small enough to fit into the capillaries to enter the capillaries. And when the red blood cell is entering, it will bend to fit in the capillaries and thus touch the capillary walls, which will increase diffusion rate. And the biconcave shape will also increase surface area, which will also increase diffusion rate. Red blood cells are also anucleated and lack organelles, which helps them carry more hemoglobin. And hemoglobin is makes up 97% of the cell, and thus picks up more oxygen to be carried by the cell. That's what we have for number 3.
Okay, before you leave that, I actually, before class, sought out, let me close this, sought out a video to show you of blood flowing through a capillary. And I'll try and make it bigger, full screen maybe. Hopefully you can now see this. So this is in a frog, and these are the red blood cells here, which you can see already within the capillary are having to somewhat contort themselves in order to be in the capillary. So let's just play its very short little video of blood flowing through. Watch, a single cell, and it has to change its shape as, say, it bumps through this narrow area of the capillary. And then it re-establishes its shape as it goes through. And you can see how they're touching the edges of the endothelial wall, which are here. Okay, let me escape that and go back to stopping the share. And I'm trying to use that video to impress upon you that blood cells are not, as you might imagine if you were just thinking about it, just flowing through water through a free pipe. They're actually having to kind of contort themselves, just like Manri said. And they also have to be small because their diameter of 7.5 nanometers is barely smaller than the diameter of these capillary beds. So an average cell is something on the order of 20 nanometers in diameter. So these are less than half of, say, an average cell in your body. Clearly, there's much larger cells and there's much smaller, but if we average them all together. So she said that's going to help in diffusion. So the shorter the pathway for the gases that are moving by simple diffusion.
The major purpose of the circulatory system is to bring oxygen and nutrients to body tissues and remove wastes.
Let me get rid of that. Okay. I didn't stop. The YouTube goes on and on, So when you're trying to diffuse the gases, and they are going to move by simple diffusion, the shorter the distance that they have to diffuse, the more rapidly you can go, as we'll learn in the respiratory system, to an 100% equilibrium. They're going to be exchanging these gases to equilibrium, and they're going to do it fairly fast. So the more features you can have to help that, the better. So how about Manreed or anybody else in that group? When you talked about them biconcave, helping the surface area and helping diffusion, did you discuss a little bit why that biconcave shape is helping that? I know you might be talking and be silenced, so don't forget to unmute yourself there you go.
For my group, we didn't go that in depth about the biconcave shape. We just knew that it'll increase surface area, which will increase the diffusion rate.
Yes, and that's a surface area to volume ratio. So the more surface area to volume, the greater the rate at which diffusion happens, just like you were saying. So that biconcave shape, you might want to say, well, if I wanted a lot of surface area, why don't I look very, very, very folded? That would increase my surface area a lot. But you also need to flex and reestablish that shape. So by being in this biconcave disc-like shape, they can not only have more surface area by having those indentations, increase the surface area to volume, but they can rebound. Now, in order to do this rebounding and reestablishing the shape as you saw them contorted, they have a particular cytoskeletal element. It's an intermediate filament. Did you talk about spectrin in your group? No?
We didn't talk about it, but I remember it from the lecture, kind of.
Okay. So if you recall, there were cytoskeletal elements that the one we talked about the most in 103A that was an intermediate fiber was keratin. And keratin had a lot of rigidity so that you had structural strength to the cell. This time we have a different intermediate filament, and it's called spectrin. So like, if you recall, keratin bound to desmosomes. Well, these aren't going to bind to desmosomes because red blood cells are not bound to any other cell. They have to not be bound. They have to be free-flowing, but this intermediate is bound to a membrane protein. So it acts kind of like scaffolding within the cell that is going to be flexible scaffolding. So think of maybe bendable metal rods that easily act spring-like to pop the cell back into its shape if you compress it. So as those red blood cells went through that narrow little area, they're getting compressed to get through, and that is allowed by the spectrum. But as soon as they have more space, they're going to pop back into that biconcave shape, and that biconcavity allows them to have that sort of flexibility working with this protein called spectrin. Okay, so they're flexible because of spectrin. They're strong because of spectrin. So think of them, as I said, like little spring-like or flexible rods that go through the interior of the cell. And then, as Manreet brought up, they lack organelles. So that is going to be for a couple reasons. You want to pull out all your organelles because you're trying to fill yourself, as she mentioned, by volume. 97% of the cell is hemoglobin. So these are just little packages of hemoglobin. 97% by volume means you only have 3% of your volume for everything else in the cell. Well, that includes the cytoplasm. There's not all that much cytoplasm. So you don't you don't want to take up rooms with things like nuclei or Golgi bodies or, and significantly, or mitochondria. So what do mitochondria use up in doing their job? Anybody?
Oxygen.
Oxygen, yeah. So these cells use no oxygen. They carry a tremendous amount of oxygen, but they don't use any of it. So they're the perfect delivery system because they're going to deliver all of the oxygen that they're carrying. Well, that's not totally true, but they're not going to use any of it. Excuse me for a second. So no mitochondria, no nuclei, hardly any space for even cytoplasm. They do make ATP, and this is going to be more significant in our respiratory system because they make it anaerobically, that is through fermentation. And at times they're going to make a waste product, which we'll talk about when we get further along in the respiratory system, called BPG. That's going to help with the unloading of the oxygen from the hemoglobin. So these are pretty amazing cells. And I would like it if even though this lecture question says list, so list, if it's list in a short answer on the test, it's fine if you just put 1, 2, 3 and name things, because generally that means it's only going to be worth, if I say list 10 things and the question is worth 10 points, then you'd get all points just by naming them. If you're writing this into the essay, which the essay I don't believe says list, it says describe. That means giving some reasons why and not just listing what they are. So it doesn't use any oxygen. It's anucleated. It doesn't need to use much ATP because it does very little synthesis. It's just managing to keep itself alive. But unfortunately, that means that it can't repair itself. So as it's squeezing its way through these capillaries, there's friction involved. We know that because remember that was part of the problem with friction causing resistance to blood flow. And especially in these tiny capillaries, they're getting deformed and rebounding, and they're going to be damaged. So the consequences of this is that they only last a little 3 to 4 months. And then they're too damaged and have to be recycled. Sorry, my bird must like red blood cells too. Okay, 97% full of hemoglobin means that each red blood cell, and these are just some amazing numbers, can carry 250 million hemoglobin molecules. Since each hemoglobin, as we'll hear more about shortly, carry 4 oxygen. That means each red blood cell, when it's in the lungs at least, has 1 billion oxygen molecules inside of it to deliver to your tissues. Okay, so let's talk a little bit more about the hemoglobin molecule, which is also really impressive. Are you going to still be the spokesperson, Manreet, or you want to pick someone else? Or did you pick someone else for that question?
No, I can just go again.
Okay.
Okay, so for number 4, I put the quaternary protein hemoglobin allows for a shape change because the bonds are weak between the double unit protein, which allows for movement to occur based on the other heme group positions. So some factors that change the shape are pH, which affects the treasury structure, and also the number of bound oxygen molecules, which is attached to the heme group. So once the oxygen is attached to the rest of the hemoglobin structure will adjust to accommodate for it. Temperature will also affect hemoglobin, as an increased temperature means an increase in mobility as well. Another factor that affects shape is the type of hemoglobin, whether it's fetal or adult. Fetal hemoglobin has a higher affinity to oxygen to ensure that the fetus gets oxygen from the mother. And we also said that BPG is another factor of shape change, but we weren't exactly sure what it encompasses.
Okay, great. That's great. You guys did a good job. So I did make a little, let me turn it on here. Okay. I did make a couple little figures to help with what she was describing, and also to kind of remind you, oops, I did the wrong thing with the share. Okay, let's do share screen again. There we go. And me talking and I don't know why it has to do that. Okay, here we go. Okay, so this is stuff that you probably had in Chem 109, general biology maybe, but just to remind you that that all proteins have, let me use a different color, that all proteins have these levels of folding, and only rarely are proteins in a quaternary structure. So all proteins have peptide bonds. These are a covalent bond, and that means they're not going to be easy to break or move. Covalent bonds link the core part of amino acids to adjacent amino acids. All proteins also typically have parts of it folded using hydrogen bonds. Now, one of the things that Manreet brought up is that hydrogen, which is pH, right, can influence these. There are so many of these because the hemoglobin proteins or the globin proteins are very, very large. So the pH changes that you see may influence the secondary structure and therefore help change the shape of the globin. But there's so many of these H bonds that we aren't going to completely denature this under normal fluctuations in pH within your blood. Now, these are arranged in two forms, beta-pleated sheets and or alpha helixes. The tertiary structure can have covalent bonds if there are sulfide-containing amino acids, but otherwise H bonding again, so that means pH can influence here too in the globin molecule. Now, these guys are held together so there are 4 globins, and those 4 globins, there are 2 types, and that's what Manreet was telling you. There are alpha and there are beta. They're not the same as the alpha helix. Sorry, they use that term a lot redundantly. So these are alpha globins, and there's 2 of those. And there are 2 beta globins too. These 4 come together using weak charge interactions. So that means you've got if we use 2 colors, you've got 4 of these things kind of weakly stuck together by, say, negative and positive attractions between parts of these huge proteins. And these things can shift and change their relationship according to conditions. So some of those conditions might be a change in the structure of the globin when H bonds are disrupted by pH. It could also be if you have oxygen binding it, because if this is hemoglobin, these globins hold 4, I put it over here, 4 heme groups. And we're going to go more into this in the respiratory system. That's a 4, sorry. 4 heme groups. And those heme groups are sort of held like a little gem in the middle. So you have a heme group in each one of these, in the middle of these. And as oxygen binds to the heme group, it's going to change how these four are related to each other as well. So here's another influence. So another thing that can affect this, because these are such weak interactions, is temperature. Because temperature makes things move, and as these guys shake, they can change their relationship again because they're only very weakly held. And then finally, and you don't have to know what this stands for, BPG, and as I mentioned, that came out of anaerobic waste product from the red blood cell itself. So red blood cells do fermentation, and the pathway, the chemistry of the fermentation changes when there's high oxygen around the red blood cell and it's saturated with oxygen versus when there's low oxygen. So when you're in the lungs and there's lots of oxygen around, it makes very little BPG. And if it has poor oxygenation, then it makes low concentration of O2. It's going to make BPG. Okay, so hold on to this stuff, because when we go into the respiratory system, then you're going to be talking about this more extensively. So lastly, she brought up fetal hemoglobin, and I actually don't off the top of my head know which of these has changed, the alpha, or the alpha or the beta, but you end up with a similar molecule, but a slightly different type of globin is making it up. And affinity is something that she alluded to. So affinity is the ease with which oxygen to the heme group. So you could think of it almost like stickiness. How sticky is it? So what we're going to see when we do respiratory system in more detail is that hemoglobin is going to be very sticky to oxygen, to the 4 oxygens it carries under certain conditions. But once we get out into the tissues, let's pick muscle because that's the easiest one. We're going to talk about in muscle, if you might remember this molecule, myoglobin. So this has a higher affinity, this has a lower affinity, and oxygen is going to jump from the hemoglobin to the myoglobin because of this low affinity, but also because of a gradient. So there's going to be a gradient, and it's also moving from high to low affinity. If you're not muscle and don't have myoglobin, not to worry, because there are cytochromes in your mitochondria that then bind that oxygen, so they have even higher affinity. So that's how you transfer your oxygen. It's moving on a gradient, yes, from the blood into the interstitial fluid, but once it's getting into the interstitial fluid, myoglobin tends to be like a magnet calling it. It's going to enter down its gradient and then glom onto myoglobin. As that myoglobin gets loaded up and you start using oxygen during metabolism, the sites on the cytochromes become available and it jumps in here and gets eventually to allow you to make ATP and becomes water. So what about this fetal thing? Let me change colors and have something else. If we have fetal hemoglobin, then what we have is, this is fetal hemoglobin. We have mom's hemoglobin over here, which has got its oxygen on it. And you're just adding one more step. And what this is the placenta. So mom's hemoglobin runs through capillary beds in the placenta that then encounter fetal hemoglobin in the baby's blood and transfer their oxygen, because this would be in this whole scale that we have, this would be the lowest affinity. And then eventually that fetal blood would encounter myoglobin or cytochromes and transfer the oxygen as you go down this affinity scale. And if you've had a baby, you might notice that long before you've really gained any weight or show at all, you tend to get breathless more easily. So if you exercise, you tend to feel like you're breathing harder. And you are, because just get used to it. The baby gets it first. For the rest of until they're adults, they're going to get whatever you're carrying first, and they're going to steal that oxygen more effectively away from your hemoglobin than your tissues can do it, and therefore, grab hold of that oxygen. So that is this whole topic we're going to revisit. We're going to look at this graphically when we do the respiratory system. So keep hold on that. All right, let's, I don't think I had anything else in here. Let's stop sharing. And we're going to go to group B. And they're going to be talking about another aspect of blood, and that is how you generate blood and where you generate blood. So there are 2 questions for Group C, and is there a spokesperson?
Hi, Professor. It's me. Stacy.
Stacy, okay. Is that a lemur? What is that?
I don't know.
I don't know what is fanciful. Okay.
So hematopoietic stem cells generate myeloid stem cells and lymphoid stem cells. The myeloid differentiate into 3 myoblasts and 1 monoblast, and eventually the 3 myoblasts become the granulocytes, and the monoblast becomes the monocytes, and the lymphoid become the B and T lymphocytes. The sinusoidal capillaries reside next to the red bone marrow, which is where the hemopoiesis happens inside the red bone marrow. And that's what I have for the number 6.
Okay, so number 6, it asks about sinusoidal capillaries. So what does that mean again? I mean, just reviewing that for reviewing purposes.
Well, sinusoidal capillaries are the most permeable, and they do happen to run next to the liver, the bone marrow, and the spleen.
Okay. So they're permeable because there are big gaps, right? So if you have an endothelial cell, there's some big gaps between them. And then they also even have some huge sinus. It's like their fenestrations got bigger too. Why is there a chirping going on? Anyway, won't worry about it. So I'm going to kind of draw this in a second without worrying about which is sinusoidal. So let me share the screen again. And just somewhat review what she, sorry, it's me delaying things. Okay, so I'm trying to draw here are the endothelial cells, and what is going to be sitting on the outside, this is we'll put the blood cells in here. This is going to be blood in here flowing through this. And sitting on the outside of this are going to be these stem cells. There's populations of stem cells sitting on the outside of this in the red bone marrow. So this is the red bone marrow. Now, one of the things you might remember with stem cells is that a stem cell, when it divides, it generates typically what we've talked about when we talked about the skin, is it generates one cell that then goes on and differentiates and usually becomes amitotic. And the other one goes back and remains a stem cell. Well, we're going to do the same thing here. And she has correctly talked about this. There are 2 names for this hemocytoblast. It's also called a hemopoietic stem cell. I actually kind of prefer that name, partly because you can abbreviate it. So do I remember now how you abbreviate hemopoietic stem cell? I think they abbreviate it like that. But at any rate, either name I'm good with. Now, unlike other stem cells, this stem cell gives rise to not just itself, so it does do that. When it goes through mitosis, it replenishes your stock of hemocytoblasts or hemopoietic stem cells, but it also gives rise to a daughter cell that is differentiating, but still a stem cell. So as Stacy, it was Stacy talking, I believe. As Stacy mentioned, there are two great lineages of these second stem cells. One of them is the myeloid lineage and the other the lymphoid lineage. Now, I put in blue here something that she started to talk about that we're going to come back to after the exam. The blue are white blood cells. So all of the cells that she talked about, the granulocytes, the agranulocytes, these are all white blood cells. We're taking that part of the chapter and doing it after this exam. Where I wanted to focus on were the further stem cell populations that these can divide into that make either red blood cells that we'll talk about now, or later we're going to talk about another group that make the platelets. So that is really the answer in a diagram to the first part. If I remember, let's come back to this because that question is also asking why sinusoidal. Well, once this bone marrow stem cells generate a cell, that cell has to make it through. So a cell or formed element has to actually be able to get through here. So you have to have a big opening. But in addition, most of the time, large proteins cannot make it through the endothelia either. And what we're going to see is we're also going to have to move large proteins from the blood. And what proteins I'm talking about are things like transferrin. Transferrin holds on to iron and is moving iron through the blood. You have to supply those stem cells with the key components you need to make hemoglobin and to make add iron to that hemoglobin. So there's a couple reasons why you want it to be a sinusoidal capillary. Okay, so the next part of the question, are you still going to be the spokesman? Goes
yes.
on to follow this myeloid, right?
Yes.
Yeah.
So it starts off with the myeloid system, which is the proerythroblast, also known as the committed cell. It's mitotic and it forms many of the free ribosomes, and it eventually turns into a basophilic erythroblast. And in basophilic erythroblast, there's ribosome synthesis. Basophilic turns into polychromatic erythroblast, and there is the beginning of hemoglobin accumulation. Polychromatic erythroblast turns into orthochromatic erythroblast, and this is where the nucleus degenerates and it's ejected to form a reticulocyte, which is where it loses most of the organelles. It becomes biconcave and is released to the blood. It still has some ribosomes, but eventually it matures into an erythrocyte, and after 2 days circulating in the blood, it loses the remaining of the organelles and it joins the other cells.
Great. Now you'll notice that as I took notes on what she's saying, I didn't go through all of the various names of the various erythroblasts. So to make this a little bit easier for you, what I'd like you to do is know the events that are happening here without knowing necessarily distinguishing the polychromatic, the various types of erythroblasts. So the events she did brilliantly. She talked about this guy being largely mitotic. So what it's doing is making more of itself, but it's also building up as it goes from the proerythroblast into the erythroblast. It's building up ribosomes. So it's making lots of ribosomes. Now, why? Because what it's going to eventually do is make a lot of protein that stays internal to the cell. If you're making proteins as a cell and you're going to ship those proteins out of the cell, you make it on the rough endoplasmic reticulum. If you're going to keep the proteins within the cell, then you're going to just do it on free ribosomes. So this cell is filling itself up with the free ribosomes. Those free ribosomes start making protein, and the protein they're making obviously is going to be, mostly hemoglobin. And eventually, as they get to a certain percentage, again, I don't expect you to know that percentage of hemoglobin, they're going to become what's known as a reticulocyte. So the reticulocyte is biconcave because it's going to eject its nucleus. And it's the ejection of that nucleus and many of its organelles, but not all of them. Sorry, I ran out of room here. That make it into a reticulocyte. Reticulocytes are where they leave, then they go to the blood. So what's crawling through when I did my little thing here is that cell, there is a reticulocyte that is crawling through here. The reticulocyte has filled mostly with hemoglobin, but it has not yet really constructed the spectrin, the intermediate filaments that are going to help keep it in the biconcave shape as it begins to circulate. It also still has some organelles. They don't say which, so that not everything has been ejected yet. Now, why that is important is because you can look at blood samples like you'll be doing in the lab today. You won't be expected to see reticulocytes. But when you look at red blood cells and you stain them with the stains that we'll be using, particularly something called a Wright stain, if there are organelles inside that reticulocyte, it's going to have some blue color to it. Otherwise, it looks kind of well, in our slides, some of them are so old they look a little bit gray, but it'll have this grayish red color that's staining the hemoglobin. But organelles pick up a different color and they're visible. So even though they're biconcave, you can tell that this is a reticulocyte when you look at a sample. Now, clinically, why this is important is that you should have, and I forget the percentage, it's probably given in the book, a certain small number of reticulocytes all the time in a sample of blood. I don't remember what the number is, but maybe it's, 3% of your blood or something is reticulocyte all the time because you're constantly going to be making new red blood cells and you're going to be, the damaged ones are going to be removed. So the new ones you're replenishing them by are reticulocytes. If that is abnormally high in a patient, that is suggestive that this patient is losing red blood cells at a faster than normal rate. So, for example, let's say you had a patient with a bleeding ulcer who was unaware of this. That blood would be being bled into their abdominal cavity, and then it would not show up as the person wouldn't be aware that they're bleeding necessarily unless they're looking at their feces or something. So they'd be unaware of it, but a blood test or looking at a blood sample from this person may show high numbers of reticulocytes where the body is trying to compensate. So how does a reticulocyte and a red blood cell differ from one another? Well, the reticulocyte still has, maybe I'll do it in a different color again. So its spectrin skeleton is not set up. It's not that it doesn't have the spectrin, and it has some organelles. We won't worry about which ones. Not a nucleus, though, and not large organelles. So by the time it becomes a red blood cell, which is happening as it's circulating, then that red blood cell has 97%, remember, by volume is hemoglobin. And it's got this mature spectrin skeleton. And it's lost those remaining organelles. So the only thing you find is sometimes some ribosomes will be inside a red blood cell, but all the other normal organelles would all be gone. And when you go to stain them, you wouldn't be able to see anything that's that blue color that you would see in the reticulocytes. All right, so by the way, it takes about 1 to 2 days for that reticulocyte to become a mature red blood cell. One other thing, which we're going to get to, I think, in the next group, is that you're making these guys at an ungodly rate. So I think it says, if I remember rightly in the book, and I don't know how accurate, but we're in a ballpark here, that you're making 2 million red blood cells every second. So every second of your life, you're making 2 million red blood cells. That hemopoietic stem cell up here, the hemocytoblasts, making the myeloid, making the proerythroblast, most of the cells that are being made are going to be going along this pathway. So it does make these others. And we're going to come back shortly and look at all the various signals that this mother of all stem cells and even these lower down stem cells have to be listening to, because they have to decide with each division, am I going to make a myeloid stem cell, a lymphoid stem cell, and then if I made the myeloid, am I going to make a proerythroblast? Now, they're always going to there'll be somebody making some of these at any one point in your bone marrow. But according to signals that we're going to talk about called colony stimulating factors, I'm sorry, the abbreviation looks like something in the brain, but it's colony stimulating factors. It'll decide which way it's going to go. Now, this problem, which we'll look at after we come back from the exam, arises when you have, say, a cancer on one of these white blood cell lines, like you're going to be drawing when you come back. That means that cancer is pulling resources to generate large numbers of cancerous white blood cells. When someone has leukemia, that's what they have. And that means you can't have as many resources to make this, and leads to a low red blood cell count in these patients, to the point where in terminal levels of leukemia, if the person was bleeding, what would come out would be like a pink, clear solution that would only have a slight pink color, not what you normally see as blood, because you're all stemming from these same original cells, and when one lineage is pulling all the resources, the other lineages can't make them. So again, we're going to come back to this diagram and look at the white blood cells. We're going to shortly look at this megakaryoblast that makes platelets. But these other things in blue, we're going to look at when we come back from this test and start white blood cells. Okay, so group C, right? Let me stop sharing this for a moment, and let's see who is the spokesperson for group C.
That would be me.
Okay.
So question 8 is, give 3 reasons why hypoxia and what is hypoxia. Hypoxia is when there is inadequate oxygen delivery to body tissues, and anemic hypoxia is when there is poor O2 delivery from too few red blood cells, or the red blood cells contain too little hemoglobin or abnormal amounts of hemoglobin. And then for question 9, describe the negative feedback loop.
Before you go on to 9, there are other conditions that could lead to hypoxia. The conditions you gave would be if you stayed in the same place and your body is in the same normal physiological conditions, and then you had some abnormal ones, like you mentioned, you couldn't make enough hemoglobin because maybe, there's a lot of reasons you can't, but you're doing a lot of mitosis. So like you mentioned, you could be vitamin deficiencies, in particular vitamin B12. We're going to talk more about this in lab today. Or you might have a lack of iron, so you can't load your hemes with iron. Or you might have, vitamin B12 is for mitosis, so you can't do enough mitosis. But what about conditions that might demand more oxygen, like I was talking about, that you got out of breath chronically? We were just talking about a condition that would get you chronically out of breath if you had it, and it would last about 9 months. That's a hint. Anyone could jump in. Pregnancy, exactly. So there you have a chronic higher oxygen demand. Or if I moved you to high altitude, and again, we're going to do this in the respiratory system more. If you go up to high altitude, then it's harder to get oxygen to load your hemoglobin fully with oxygen. So, and then finally, you could have something that's wrong with your respiratory system. Maybe you have pneumonia and it's hard, or you have, God forbid, COVID, and it's hard to get enough oxygen to the hemoglobin, even though you have enough hemoglobin. Those are all complicated reasons why you could end up with low oxygen tension in your tissues and therefore have hypoxia. So good. So let's go on to 9 now. I think it was Noah.
Yeah.
And talk about how that triggers a negative feedback loop. And maybe what I'll do is share the screen again, and I'll just draw it as you're saying it so people can see things. I gotta wake this up again.
Okay, so do you want me to wait till your screen comes up, or?
Oops, sorry, I forgot to trigger it. Okay, so go ahead.
So the first is the stimulus, and that's the hypoxia. And then the kidney, kidney is like the primary detector here, but the liver is also can release erythropoietin or EPO.
Mm-hmm.
And then erythropoietin will then stimulate red bone marrow to enhance erythropoiesis. And since there is an increase in erythropoiesis, there's more red blood cells. And then therefore the oxygen carrying capability in blood is increased. And that will negatively feed back into the original issue.
Okay, so the only thing I'm adding to that is that it not only makes more red blood cells, it speeds up the time it normally takes is something, I think the book says like 45 days to go from the myeloid stem cell to a red blood cell. And it's going to make that go faster. So the rate and the amount of red blood cells produced, and then that will mean that you get an increased oxygen carrying capacity. Did I spell that right? And that will then be the negative feedback. So hopefully it means your hypoxia goes away and you don't need to do this. But what are the consequences of having, let's say you're a normal person with normal blood and you are suddenly made to live at high altitude for the next 3 or 4 months, or maybe longer, 6 months. what's going to happen to your blood? You're going to have red blood cells produced, right?
You'll have more because of the different conditions, right?
Yeah, and that means what feature of your blood is going to get changed? That
The
might
viscosity?
Viscosity is going to go up, which means you're going to have to raise your blood pressure chronically in order to push that thicker blood through your vessels. Luckily, women, you don't see a big change in that one. Usually you can manage to keep your tissues from going into hypoxia enough that when you're pregnant, you're not going to get higher viscosity blood, but it could happen at altitude. And people who have evolved at altitude, lived many, many generations at altitude, have compensated for this. So, Tibetans on the Tibetan plateau, that sort of thing. Athletes that then compete. So let's say you are an athlete that normally is a summer Olympian, and you're swimming or running or whatever you're doing at sea level. And then, like when they had the Olympics in Mexico City, you have to go and compete at a higher altitude. They typically will go and live at that altitude. If they're doing it without blood doping, they're going to go live at that altitude for long enough to raise their red blood cell count. But of course, then they're challenging their cardiovascular system, which hopefully is plenty to meet the challenge by having thicker blood. And then they're, of course, doing their thing, which may challenge their cardiovascular system even more. Okay, so that's great. And the next question has to do with generating this blood, right? Let me stop sharing again. So question 10, I think you're in the same group, right? No, maybe not.
That'd be our group, actually.
Yeah, that is the next group. Okay. So who wants to talk about the question 10?
I'm going to go ahead and cover question 10. So for the 3 iron binding proteins, we have ferritin, hemosiderin, and transferrin. Ferritin and hemosiderin are both proteins which the iron binds to in the liver primarily, as well as the spleen. And this is because free iron is toxic, so it needs to be binding to these 2 proteins. Transferrin is either an alpha or beta globulin, which iron binds to when it's being transported out of the liver spleen into the red bone marrow.
Okay, so to summarize, we had 3 iron binding. Now, why you need to bind iron is because iron is very toxic. Remember, iron has 3 positive charges. So that means it's very good at disrupting things that are partially negative, partial negative charges on it. It's a toxic substance. So in order to keep it from being toxic to your body, you need to bind it up. Now in hemoglobin, it's bound to the heme group. So that detoxifies it as a way, way of detoxifying it. But if you want to move it through the body, then you need to move it on transferrin, which is one of the, and again, I probably should learn, but I haven't learned. It's one of the alpha or beta globulins. So you might remember back when we did blood plasma, we talked about alpha and beta globulins were making up nearly 40% of the protein in the blood. One of those is transferrin. It's not making up 30 to 40%, but it's one of the ones that is made. Like most of all of these alpha and beta globulins, the liver is making it. Now that is convenient because, as Sana told you, the liver is where we're going to store the iron. So this is for transferring to transfer to, to move the iron. You use that. But you need to store it because remember, you're doing this 2 million cells per second, each cell with 250,000 hemoglobins in there, 250,000 heme groups in there, I should say, in order to bind up 1 billion oxygens. So you're asking for a lot of iron to make red blood cells. So iron is not super available in your nutrition and what you take in your body. So if you come across a source, especially as we evolved, you're going to store as much as possible. So you're going to store it in the liver. You're storing it. And the primary way to store it is stored, on ferritin. If you got so much iron, so this is the primary storage molecule. And typically, probably with normal diets, you're just storing it on ferritin. But if you come across a lot of iron in your diet and you want to store it, there is a secondary storage molecule called hemosiderin. So you can see the word heme in there. I don't know if it has a heme group, but it's the second way in which you can store iron when you have plenty of it in your liver. So one good source of iron in your diet is to eat liver. It's storing it so you can get lots and lots of iron if you eat liver. Okay, that's great. So the last part, which is question 11, is saying, well, let's talk about how the red blood cell, or the fate of all the products of the hemoglobin when the red blood cell is broken down. But maybe you could start with where we're going to break all this down. Did you cover that, Sana, or?
Professor, it's Jasmine. I'm covering this question.
Okay. Oh, you're on that question. Sorry, Jasmine.
Yeah, no problem. Okay, so describe the fate of the hemoglobin's molecule when it's recycled. So I put down the damaged red blood cells are largely detected in the spleen, liver, and bone marrow. Mm-hmm. They detect it through their sinusoidal capillaries, and the macrophages on the outside of those capillaries look for the damaged red blood cells. Right. So the macrophages reach through, grabs the red blood cells, and pulls it out of the blood, bringing it into the interstitial space where they engulf it. They then take the hemoglobin and separate it into its protein parts, which they then separate it into its amino acids. The amino acids are released into the blood from the liver or the spleen. The heme group is separated into iron, and if you're in the liver or the spleen, it'll store the iron into those ferritin or hemosiderin proteins. The rest of the heme, such as the carbon, oxygen, hydrogen, and nitrogen, are taken apart and made into bilirubin by the liver, spleen, or bone marrow, which will then be released into the blood. The liver takes the bilirubin and turns it into bile, and the kidney converts it into urochrome, which is what makes your pee yellow. The bile is secreted into the intestines to digest the fat. A lot of bile is retaken into the digestive tract and recycled. So some of the bile makes it into the small intestine, into the large intestine, where sorry.
I know that's the problem.
Where was I? So it makes it into the small intestine to the large intestine, where the microorganisms can convert it into sterocobalamin, and the sterocobalamin makes your feces black or brown. That's what we have.
I forget how to spell this one. I had two. I can't. Something like that. And then the kidney, it's made into urochrome, you said, right?
Yes.
And that means that's the yellow color of your urine.
Yes.
So now you know why your pee is yellow and your poop is brown. It all has to do with bilirubin. So bilirubin is in your blood because it's being moved from the spleen or the bone marrow, and these are all doing the same thing, towards the liver. as you may know, if you don't adequately get rid of it, bilirubin itself has a yellow color. So it has sort of a yellow hue to it. And in folks that aren't able to get rid of it adequately, so if that was an adult, it might be somebody with liver failure, because after all, most of that is either leaving through your kidney, but most of it is being recycled as bile, made into bile. You end up with a yellow color, which is called jaundice. So jaundice, if I write that right. Jaundice is also not uncommon to have in newborns because their liver has not, well, you're not doing all the functions that the liver does normally when you're in the womb. So the liver hasn't matured enough to process the bilirubin. Plus, when you're in the womb, you're still making red blood cells and you're unable to totally recycle those blood cells. So as we'll see when we do the digestive tract later, a lot of it is dumped into your stomach, and it makes the first set of feces that called meconium that the baby passes. That's a whole lot of unrecycled red blood cell parts, including the heme groups. And the babies are sometimes born where their liver is too immature to process that amount of bilirubin, and so they have jaundice. Apparently, UV radiation helps them break it down, so they're often put under UV radiation, low levels of UV radiation to help them break down their yellow excess bilirubin until their liver gets really up to gear and gets rid of it. So this is another theme that's going to be coming back throughout the semester, as later we'll look at the roles of the liver and what happens when we do the kidney, what are the components of urine, and urochrome will come back to us when we do the kidney later. So worth hanging on to all of this. Why do we have bile? Does anyone, just to throw that in? What is bile needed for?
Fats.
What did you say?
Digestion of fats.
Yeah, it helps. It's not an enzyme itself, but it's going to help us be able to digest fat in the amount of time that the food is passing through the small intestine. And it also means that fat-soluble vitamins are going to be better able to be absorbed if you have sufficient amounts of bile. So bile itself is made by the liver to do that. Most of it gets recycled back to the liver, but some of it makes it past the liver. And then microorganisms in your large intestine convert the bile into this substance. Okay, let's move. Let's see how much time. Oh, we're almost done, huh? So we probably won't quite get to the platelets because that's our next topic. We only have a couple minutes left, or a minute left. So let me just check chat before I get too far away here. Okay, doesn't look like there's anything critical. So when we come back, Group E will speak, and we only have, I think, a group F and G. We'll have 3 groups to go through hemostasis. And I mentioned this last time, but I'm just going to reiterate here that I've really edited this. So make sure that when you're studying this, when you're writing up your lecture questions and your essays, you don't have to explain all the Roman numerals and every single aspect of, especially where it's edited, is the coagulation part. So use the PowerPoints as a guide to how much you need to know about that. Okay, so I'll see you in lab in a few minutes. We're going to be working on a blood lab today. So make sure you have the handout for the blood lab. And I will see you guys, or most of you, in about 15 minutes. All right, I'm going to stop sharing and I'm going to end here. And if you can just show up with that, we'll see you shortly. Okay, bye.